Students, now let us study the very first point of our syllabus, that is from cell biology. So in this point we'll learn about the cell, general structure of the prokaryotic cell and eukaryotic cell. So first of all, here is an introduction of this topic. uh, See, all the organisms we know, they are composed of cells. So if you take example of our houses which we live in, the houses are made up of number of bricks. In the same way, all the living organisms are composed of cells, and these are of different types of cells present in our body or in each living organisms that we know. And among these all organisms, some organisms are unicellular and some of them are multicellular. Unicellular, we know, uni means one or single celled organisms, and these organisms perform all their bodily activities or all the functions independently, whereas multicellular organisms perform the life functions depending on each other. Anything less than a complete structure of a cell doesn't ensure independent living, and therefore we consider the fundamental structural and functional unit of all living organisms, that is a cell. So here all living organisms are composed of cells. Unicellular means single celled, multicellular means many celled. Prokaryotic cells basically means primitive type of cells, okay, and eukaryotic cells, if you compare with the prokaryotic cell, they are of advanced type of cells, okay, evolved type of cells. So prokaryotic cells basically they have membrane bound nuclei. Eukaryotic cells, they have membrane bound nuclei, whereas cells that are lacking a membrane bound nucleus, they are called prokaryotic cells. In both prokaryotic and eukaryotic cells, there is a semi-fluid matrix called as cytoplasm. So cytoplasm is a semi-fluid matrix present in all the organisms, including prokaryotic and eukaryotic cells, and which occupies the volume of the cell. This semi-fluid is occupying the volume of the cell. So besides the nucleus, in eukaryotic cells, they are having other membrane bound distinct structures. If you take example of eukaryotic cells, in which there are number of cell organelles which are membrane bound structures like endoplasmic reticulum, Golgi complex, lysosomes, mitochondria, then microbodies, other like vacuoles. So all these are membrane bound structures only present in the eukaryotic cells, whereas in prokaryotic cells there is a lack of such membrane bound organelles. So primitive type of cells are prokaryotic cells in which there is no membrane bound organelles. Examples of the prokaryotic cells are bacteria, blue-green algae, mycoplasma, etc., etc. Okay, and one more important thing I want to tell you here is that the ribosomes, which are non-membrane bound structures or organelles found in all the cells, eukaryotic and prokaryotic cell, ribosomes. Ribosomes are non-membrane bound organelles found in both the eukaryotic and prokaryotic cells. And another cell organelle in animal cell which is also a non-membrane bound is a centriole, and we know this centriole is helping in the cell division process. Okay, so this is the introduction of the chapter. Now let us move to the prokaryotic cells. So the prokaryotic cells, we take example here as a bacterial cell, which is a typical example of prokaryotic cell. These vary in shape and size. So prokaryotic cells as a bacteria which we see with a number of shapes and sizes, like bacillus shape, which is a rod shaped, coccus bacteria, that is spherical in shape, vibrio bacteria, that is comma shaped, and spirillum, which is a spiral structure. So if we see the bacterial different types of bacteria, we can observe their different shapes and sizes. So all the uh, prokaryote, prokaryotic cells, they have cell wall surrounding the cell membrane, and they have cell wall surrounding the cell membrane. The fluid matrix filling the cell is the cytoplasm. There is no well-defined nucleus. Well-defined nucleus is absent in these cells. Then genetic material is not enveloped by nuclear membrane. This is very important point to be noted. Genetic material is not enveloped by nuclear membrane. And along with that genomic DNA, bacteria do have other circular DNA with it, and these are called plasmid. So two types of DNA are present. One is genomic DNA, and one more DNA is found, that is a circular type of DNA, and that is called plasmid, which is present in the bacterial cell. In a bacterial cell, there is a plasma membrane, and which is uh, around which there is one more layer called cell wall. These are the bacterial shapes of the bacteria that we have discussed just now. And this is the structure of our prokaryotic cell, which is a bacterial cell, right? So in this diagram, you can see clearly all the structures present in the prokaryotic cell. As I told you, there is cell wall, and around this bacterial cell there is cell wall, and one more, if the plasmid is present, then it is known as positive type of strain, and if there is no plasmid in the cells, there it is a negative type of strain. That we will discuss later on. But first of all, see the basic structure of the bacterial cell. And yes, we are talking about the cell wall, which is made up of peptidoglycan. So what is the cell wall is made up of? The cell wall is made up of peptidoglycan. Now outside this cell wall there is also one more capsule, and that capsule is called glycocalyx. In the after the cell wall there is one more capsule like structure called as glycocalyx. All these are the protective in function. These structures, or these all cell walls and capsule, all the structures, they protect the bacterial cell. Now this bacteria can be classified into two groups on the basis of differences in their cell envelope. Bacteria, they are classified into two groups on the basis of differences in the cell envelopes and the manner in which they respond to the stain of gram stain. The bacterial cells stained with the gram stain are called gram positive, and if they are not taking that stain, they are negative type or gram negative type of bacteria. The plasma membrane is semi-permeable in nature and interacts with the outside world. A special membrane structure is called mesosome, formed by the extension of plasma membrane. So mesosome is present, and that mesosome is another structure which is the extension of the plasma membrane. It is nothing but the extra or extension of the plasma membrane, which is forming a structure called mesosome. Now what is this mesosome or helping in This mesosome helps in the cell wall formation and DNA replication, as well as in the process of respiration. Okay, so mesosome helping in the cell wall formation, DNA replication and respiration. Then this bacteria cells may be sometimes motile or non-motile. If they are motile, then definitely they have flagella. Along with that, they can move. So motile cells have flagella and non-motile have no flagella. One more structure is present, that is called pili, and the pili are elongated tubular structures made up of protein. Okay, so flagella is there, pili are present, and fimbriae are also there, which are small bristle like fibers, and they help in the adhesion to any surface. So how many structures are present? Extensions are present. That is flagella, second one is pili and fimbriae. And the plasmid is there, some ribosomes are present, then the cytoplasm is always present in the cells. Okay, and mesosomes, which are helping in the process of respiration and also DNA replication. Okay, so the bacteria which are having the or which can be stained are called with the gram stain are called positive type of strains, and gram positive strains, and gram negative they are not uh, stained with the gram. Those are negative type of strain, gram negative bacteria. Okay, so this is about the prokaryotic cells. Okay, in the next lecture we will discuss about the another type of cells, that is eukaryotic cells.